All right, let's go ahead and get started. Before we start with the lecture, I had two quick announcements. One, we have our first draft of the figure illustration due on Thursday. Again, each of you can get a free trial of BioRender. It's a really user-friendly software that creates really compelling publication quality figures. And I think it's pretty intuitive on how it works. You can use the search bar to find certain graphics or certain objects that you can use to build your figure. For building the figure itself, we have some pretty successful examples available for you on Canvas to help guide you as you create your figure. If any of you are panicking to try and figure out how do you actually create a figure like this, the main idea is that you're trying to communicate what the problem is in your disease or biological problem and what your proposed idea is. So for example, if there's an unknown mechanism, you can kind of try and highlight what is known about this disease and what's unknown in some way, and then try and propose some specific mechanism that ties things together. If it's some specific alternative therapy that you're proposing, you could try and illustrate what's the current therapy that's used, why it doesn't work. And again, there's no single right way to try and create the figure, but rather to try and communicate it in a clear and compelling way. And to try and if it's an alternative therapy, try and illustrate what your alternative approach is and why perhaps it's even better than what's currently used on the market. Are there any questions about the figure illustration? I'll take any and all questions. Yeah. If you run out of free icons available. I didn't realize there was a restriction on the number of icons you could use within a single figure on the trial. Has anyone else run into that issue? Show of hands. What you might be able to do, the quickest solution I can think of off the top of my head is, well, one question I have you tried creating a second figure and then using icons in that second figure and then like potentially like combining them into one on like PowerPoint or something? Okay. That was just a quick thought where like if potentially you have like your one figure that has like 90% of what you need and a second figure, because I think it's up to 5 figures you can do on the free trial. You could create a second figure and then basically merge those two in PowerPoint on a slide. Give that a shot. And if you run into issues, and also give that a shot. And for any of you who didn't raise their hand but have that same issue, give it a shot. And if you run into issues, please let me know. I think this is going to be a better approach than giving everyone my single account, because we run into issues of people booting each other off the day before the assignment's due because only so many people can be on it. It's only one. For some reason. Okay. Thank you. Are there any other questions about the figure assignment? That was a great question, by the way. I appreciate you sharing that. All right. So for the second announcement, it looks like a ton of people have been using Piazza. That's great. I appreciate you guys doing that. If you guys have any more administrative related questions, whether it's about things being posted on Canvas or certain questions about slides, I'll say go ahead and email us. I'd want to reserve the Piazza discussion board for more content-focused conversations. But also what I want to say is we're doing our best to try and post these slides and post these assignments in a timely manner. Everyone has a million things going on, and we're trying to do our best to like update these slides and post things up. And again, like I think BME students are typically probably, again, I think no one will probably argue with this here, some of the most busiest students probably on grounds. But likewise, everyone on the teaching team also has like a million things going on. So everyone I think is fairly busy, just trying to handle and manage a lot of things. So I'm just going to ask that you guys give us a little patience when we're trying to get things up. So like all of the Unit 2 pre-lecture slides and lectures and videos are already posted. We're trying to finish updating Unit 2 slides to make sure that everything is communicated in a more clear manner, but also updated content-wise for what's been published in the last few years that can help, again, this is a class of half-truths. How have things been, what have we learned, and what's been identified lately that kind of help update this material. But again, I just want to ask you guys to just give us a little patience, and we're going to try to do our best to get things posted for you guys in a timely manner. Does that sound good? See gentle nodding, so I'll take that as an affirmative response. With that being said, we can go ahead and jump in. These next couple lectures are some of my favorite material, so I hope you guys are excited as I am. But we're going to be pivoting a little bit because so far we've been focusing a lot on the molecular biology side of things. We've been focusing a lot on proteins and amino acids, different classes of proteins, how they're situated within a cell membrane or within the cell cytoplasm. Today, we're going to pivot and focus more on the cell bio side of things. And particularly, we're going to be focusing on innate immunology. There's going to be two arms of the immune system we're going to be focusing on. There's the innate side, and there's going to be the adaptive side. And I have a figure that kind of helps understand the breakdown of those two arms of the immune system. But we'll be focusing on innate immunology, particularly we're going to be looking at it through the lens of Crohn's disease today. So Crohn's disease, you may have heard of it, and it may be a disease that can sometimes be easily confused with another similar disease. But Crohn's disease is a type of inflammatory bowel disease. That's a class of diseases that have these hyperinflammatory lesions within the GI tract. Under inflammatory bowel disease, there's two diseases. There's Crohn's disease and there's ulcerative colitis. As you can imagine, colitis affects the colon. Whenever you hear itis on the end, that typically means inflammation of. So colitis is inflammation of the colon. The way these two diseases manifest are pretty different, as well as the mechanisms behind them. Ulcerative colitis usually is restricted to the colon, as the name implies, whereas Crohn's disease is really the entire GI tract is susceptible to damage here. Typically, within ulcerative colitis, the mucosal layer is typically what's affected, whereas with Crohn's disease, we'll actually see lesions that affect the entire full thickness of the epithelium within the gut. And finally, this will make a little more sense when we jump into adaptive immunology, but there are certain cell populations that correspond to each of these. With colitis, it's more of a Th2 mediated disease. That's a class of T cells. And Crohn's disease is more of a Th1 and Th17. These are different flavors of T cells, and T cells are one of the major populations within adaptive immunology. So you can, if it doesn't really make sense or if it sounds like an alphanumeric soup, again, that's a lot of what immunology is, but the idea is that we're going to try and break it down and make it a little more digestible. And so those different cell populations are going to make more sense after the next lecture, but those are the key distinguishing factors between Crohn's disease and ulcerative colitis. Some of the key symptoms here, again, with Crohn's disease, it affects the entire GI tract, and it can be an entire full thickness of the epithelial layer. So as you can imagine, that's going to affect nutrient uptake within the GI tract, and which will then affect growth within people who are suffering from Crohn's disease. It can also be manifest with fever and, as you can imagine, weight loss as well. What you see here is there's actually an image of the GI tract with these abscess lesions within the epithelial layer there. An abscess is usually a pus-filled lesion, and pus is oftentimes characterized by being these dead cells of the immune system called neutrophils. They're the first cell that come into the site of infection, and they oftentimes are tasked with just eating things up and trying to break things down. But typically, when an infection persists, a lot of those neutrophils come in and die, and so pus is lots and lots of dead neutrophils. As you can imagine, with lots of abscess lesions within the GI tract, it's going to be associated with a lot of pain and discomfort. It's also going to result in diarrhea. Ulcers, where these lesions are basically like slight holes within the GI tract. And finally, in more severe circumstances, it can actually result in a fistula. A fistula is kind of like a connecting hole between two cavities. So we can have a fistula that goes from the rectum to the bladder, the rectum to the vagina, the rectum to, I'm trying to think of another common fistula with Crohn's disease. Those are the two most common ones I've kind of come across in reading. But again, a fistula is kind of a hole that connects two separate cavities within the body here. As you can imagine, there's also associated anemia because of the affected nutrient uptake, as well as sometimes associated neurological issues that can emerge as well. For the prevalence, when we look at it with inflammatory bowel disease in general, there are about 1.5 million people within the United States alone that currently suffer from IBD. Specifically with Crohn's disease, however, with the incidence rate, there's about 1 in 10 cases per 100,000 people within the US specifically. What's interesting is that there's a bit of a bimodal distribution if you look at the number of cases as a function of age, meaning that there's large peaks of patients that are within like the teenage years, as well as those who are around in their 50s. We actually don't really know why. It's not clear why there's seemingly specific ages of patients that present with Crohn's disease, but that's something that still needs to be, that's something we still need to determine. As far as causes for Crohn's disease, we've talked before about monogenic and polygenic diseases. Monogenic diseases are diseases that are a result of one single mutation in one gene, whereas polygenic diseases are a family of genes that have been affected, or a group of genes that are affected that cause this disease. Crohn's disease is a polygenic disease, and so there's a number of mutations that enhance susceptibility by causing the innate immune system to be dysfunctional here. And so there's, these genes probably fit within a class of certain gene families, but it's still polygenic because it's multiple genes. Again, the example we've talked about before that's monogenic is sickle cell, because there's a specific mutation on the beta chain hemoglobin gene, so that made it monogenic. This is a family of genes that are associated with innate immunity, but still would consider it polygenic. What's interesting is that what we talked before about the cell populations that are associated with Crohn's disease versus ulcerative colitis. Those were adaptive immune cells. It's believed that because of the dysfunction in innate immunity, there's an overcompensation of the adaptive immune system that then contributes to the disease here with Crohn's disease. And some of these symptoms that we see are also a result of continuous activation of innate immune cells to microbial infections, which then potentially explains why we see a lot of these abscess lesions form within Crohn's disease. So before we jump into the content here, I want to hit pause and give space for any questions here. Yeah. Well, so neutrophils are not an adaptive immune cell. They're an innate immune cell. But typically, whenever there's some sort of infection, neutrophils are going to be, for lack of a better phrase, one of the first cells on the scene. They're also pretty short-lived. Like, we're talking on the order of hours to... Actually, yeah, we're going to stick with hours. On the order of like a few hours to maybe 48 hours at like the most long-lived neutrophil in response to infection. Just because typically they're tasked, and we're still understanding some unique functions in neutrophils. There's some kind of going back to maybe like the ECM lecture. There are some neutrophils that have been associated with some unique functions tied to ECM and tissue repair, but particularly as it relates to infection, they are fairly short-lived, and so they'll die pretty quickly. That's typically how they function in response to an infection. That was a great question. Are there any other questions about Crohn's disease or colitis? Yes. Yeah. Actually, that's a great question. That's my impression, that typically when we talk about that bimodal distribution, I believe it's at age of diagnosis, but I'm actually going to double check to confirm. That's a good question. Are there any other questions here? I'll post an announcement in response to that, because I think that's actually a really great question. Okay, let's go ahead and dive in. What we're going to be doing today is going to be looking at one specific gene that's associated with both innate immunity and Crohn's disease. Excuse me, it's called NOD2. And so we'll put a pin there and we'll come back to it. And for now, we're going to go ahead and jump into innate immunity. And so what we have here is we have a graph that illustrates, or a figure that illustrates the different cells that emerge within our immune system. And they all generate from a hematopoietic stem cell, which is like the main stem cell in your bone marrow, that gives rise to all other immune cells in your body. And they actually branch off below here to either a common myeloid progenitor or a common lymphoid progenitor. And I should say, I'll hit pause here. This is to help explain the difference between innate and adaptive immunity. I'm not going to ask you to memorize this. Most grad students, unless you're like in the immunology program, are not going to be expected to memorize this. Maybe like one specific lineage, if like one cell is like your entire dissertation. But the idea here is that when it splits below the hematopoietic cell to the common myeloid and lymphoid progenitor, those are like the main stem cells, basically, for all the innate immune cells on the left and all the adaptive immune cells on the right. So the cells we're going to largely be focusing on today are the cells to the left, the myeloid progenitor cells that, or the cells that stem from the myeloid progenitor. And so you're going to be hearing about a few of those. But this is just to illustrate what we're talking about today is on the left-hand side. What we're going to talk about next class is on the right-hand side. This is something we talked about that was talked about in the pre-lecture, but I want to try and take a second to try and talk about this a little bit more specifically as it relates to innate immunity. One of the key qualities of our immune systems is their diversity. Our immune systems are able to respond to a vast array of pathogens. And in particular, as it relates to our innate immune systems, what I can define here, pathogens are foreign microbes that kind of invade our bodies in different ways. But the diversity of pathogens, what's really interesting here is that our innate immune cells will actually respond to specific patterns or structures on these pathogens and can respond to a wide array of them. In addition to that, there's a specificity that exists within our immune system. And this is all going to differ in degree between innate and adaptive. But the specificity here is that there's these features on these pathogens, these structures or these patterns that certain receptors on our immune cells will recognize. Certain patterns of sugar molecules with proteins, with certain sugar molecules with lipids. They're meant to try and identify specific regions that are also associated not with ourself, but with some sort of foreign pathogen. So the main function, so like if I was to like put like a huge asterisk on something here, the main function of our immune system is to identify self as self and distinguish what's foreign. And so one of the things here is that the specificity here with innate immunity is that it identifies patterns on things that are not self. As you can imagine, our cells have DNA, but they're in our nucleus. So if there's DNA, for example, in the cytoplasm, that's a problem. It's probably not from our own cells. And so there's a series of receptors we're going to talk about that identify some things. And so there's going to be moments where I'm going to say, like, here's a virtual asterisk of how our innate immune system has selected for unique receptors that recognize things that are not associated with our cells or our immune system. And finally, there is a really exquisite memory here, particularly with adaptive immunity, but also to some degree with innate immunity as well, to try to remember some of these previous encounter pathogens. The memory for innate immunity is that it's been genetically encoded and passed down throughout selection, where these receptors that recognize patterns has been genetically encoded and passed down. So that's why it's called innate. It's built in and genetically encoded, whereas adaptive, you acquire that immunity. Another name for adaptive immunity is acquired immunity. There's also a really unique segment of research called innate memory that really starts to go beyond just the idea of these receptors being genetically encoded, but rather that our innate immune cells are actually able, and again, this is just like a cool thing because I kind of geek out about innate immunity. There's this cool idea of innate memory where beyond just like what's genetically encoded, that our cells are able to respond stronger to some stimulus they've seen in the past. And there's some sort of mechanism that enables them to do that. It's the potential mechanism there is something that we're actually going to talk about later on in the semester, but innate memory is cool. I just kind of want to like drop that in there because I think you guys would appreciate that. Another way in which our immune system can be broken down is down to a humoral component and a cell-mediated component. Humoral is, you can think of that as either primarily in the blood or in some of the other bodily fluids. So we're going to break this down into a cell-mediated component and a, for lack of a better phrase, bodily fluid component, primarily in the blood, but also elsewhere. Excuse me. For innate immunity, the humoral component is something that's called the complement system. There are a set of enzymes that are meant to try and directly kill either some foreign cell or some sort of foreign pathogen. And the cell-mediated response, typically, for innate immunity is cells respond to foreign pathogens by actually engulfing them through a process called phagocytosis. For those of you who are who might recognize some of the prefix here, phago usually means like eating, and cytosis is usually of the cell. And so this is the cell eating some sort of foreign pathogen. And there are certain cells that are dubbed phagocytes here, and we'll talk more about that in a little bit. But those are the main humoral and cell-mediated components of innate immunity. For adaptive immunity, this is just a teaser because we're going to talk a little bit more about this in the next lecture. The humoral component there are antibodies. Antibodies are these proteins that are in our blood that usually emerge in response to some sort of pathogen that has a lasting memory. This is how vaccines work. We have antibodies that emerge in response to a presented pathogen, and that helps to inactivate it. The cell-mediated component are going to be specific cells that respond by eliminating whatever cells have been infected by some sort of virus, or sometimes can also be a cell that has undergone some sort of transition, potentially becoming perhaps a tumor cell. And so those are the main components here that's the main breakdown at a high level of our innate and adaptive immune cells here. I'm going to hit pause and I'm going to wait for a couple of questions. I'd love to have a couple of questions here. Yeah. Yeah, so that would be part of the cell-mediated innate immune response. So like neutrophils are really great at secreting a ton of enzymes that degrade things that literally they secrete something called perforins. It literally pokes holes in pathogens as well. So they're another great way. This is a bit of a layman's way of describing it, but I'll try and maybe make it a little more refined. Innate immunity is fast but dumb. Adaptive immunity is slow but smart. So innate immunity is fast. Like, and again, neutrophils come in on the order of hours, but all they're doing is dumping a ton of enzymes, dumping a ton of other components that will activate other cells, then they die. Macrophages will come in, they'll eat everything, they'll want to engulf it, it's a phagocyte. And so like, again, it's fast, it's recognizing patterns, but adaptive immunity will eventually kick in if some pathogen escapes these other cells. And they're going to be way more targeted on the order of like, not patterns, but like down to like amino acid sequences, and will remember it for perhaps the rest of your life. And so that's kind of the main difference between innate and adaptive immunity, and why neutrophils, for example, I keep throwing neutrophils under the bus, I feel bad. But that's part of the reason why some cells will have a short-lived effect, whereas other cells have much longer-lived effect. Yes. I'm smiling because it's a good question. Sorry, keep going. Yeah, so like this is an entire field of immunology that is also super complicated and I never wanted to get into. So I stuck on the innate immune side because I like fast but dumb. Um, so there are a lot of factors that go into vaccine efficacy and lasting duration. Like that's one of the that's been one of the discussion points with some of the COVID vaccines, where needing to promote better long-lasting immunity. And so some of it has to do with how the vaccine is developed. Some of it has to do with how it's delivered. So for example, for the mRNA-based vaccines, like a lot of them were shot in the arm, but COVID does not affect intramuscular area. Like it affects primarily our intranasal space, where there's a different kind of antibody that's produced there than what is generated here. Like we have 5 flavors of antibodies. This is just like a quick jump in where like one of the flavors of antibodies is called IgG. It's the most common one. It's oftentimes produced, but in our nasal cavities, we have another one called IgA. And so a lot of the same folks that develop those antibodies are now trying to develop an intranasal COVID vaccine that develops IgA that's actually meant to be in the nasal cavity as well as within our upper respiratory airway. And so how you deliver the vaccine, how it's designed, there's a lot that goes into vaccine development. There's a great scientist up at Yale University who's like the COVID guru, for lack of a better phrase. She's been publishing a lot on this. Her name is Akiko Iwasaki. She leads I think she might be chair of immunology at Yale. But she's publishing a lot of like specifically like vaccine for different viral infections and how we can promote or provide better vaccine development. So like, because this is not my area, I'm going to try and point you to like, I think like the leader in the field in that area who I think is doing some cool work. But I hope at least that gives you a semblance of like why there's different responses to different vaccines. Yeah.
Also,
yeah, please.
The idea of like conjugating whatever you are administering with the vaccine to something else or including different elements in that vaccine that are going to enhance the immune response. It can be things like certain elements or compounds that don't have something to do with the actual pathogen or your body's ability to recognize that pathogen, but rather just ramp up the immune response in response to the actual pathogen of the vaccine. So they can do a lot.
That's a great point. Thank you. All right, I'm loving these questions. I'm going to say in the interest of time, we're going to press on, but I'm also going to have other points where I stop for questions. So if there are other burning questions, I want you guys to keep them coming. So we're going to take a second to focus on the humoral innate component that we just previously touched on the previous slide. That's going to be the complement system. So again, the complement system, if we think for this is a slide where it is really easy to get lost on all the finer details. I want you all to first focus on the big picture of what complement is for, and then maybe focus on like the different kinds of the different pathways of complement, focus on the big picture of each of these pathways, and then you can start providing in the details. So I'm going to make sure that we focus on the big picture, get those take-home points out, but then I'll try and fill in with some of the finer details. So again, complement is meant to try and target and destroy invading pathogens or other invading foreign cells. It's this really unique cascade of enzymes that accomplishes this goal. There's three particular pathways within the complement system. We have the classical pathway, the lectin pathway, and the alternative pathway. First, starting with the classical pathway, usually what's going to end up happening is that there's going to be some sort of antibody that recognizes something on a bacterial surface or a pathogen surface, and those antibodies will bind. These antibodies are incredibly specific. In this figure, we see two antibodies there. It's possible that these two antibodies bind and recognize the same peptide sequence, or they could be recognizing different elements. They each recognize a different thing, but both recognize a different aspect of this bacterial pathogen. But the idea is that we have antibodies that bind, and there's this huge docking protein called C1q. A lot of this is going to be in numerical order, so that I hope that kind of helps get rid of some of the mystery. But C1q will come in and dock, and what ends up happening is that all of these proteins are within our circulation. Whenever there's a specific site of infection or some sort of pathogen that's present, what's actually really interesting is, and I'm sure everyone's kind of experienced in some way or another, the question sometimes can be like, if you take Advil, how does Advil end up at the right spot? Or with these circulating proteins, how do they end up in the right spot if it's circulating in our blood? Usually when there's some sort of infection or injury, oftentimes your blood vessels will get activated to be leaky at a certain location so that things in circulation go to that site. For lack of a better phrase, your blood vessels will get sticky, where immune cells can actually bind to the inner part of your blood vessel and like pass through and go to that specific site where it is leaky. There's also with inflammation you'll have, because things get leaky, there's a lot of edema, fluid leaving your blood vessel to go to that site, and that's going to impinge on some nerves. So typically with inflammation, again, like think of a bruise or think of what it felt like when you rolled your ankle or busted your lip. You're going to have a lot of swelling. It's going to get hot. It's going to get the blood vessels, will lack of a better phrase, get leaky and sticky because all of these immune cells, all of this fluid is going to that site, but it also enables some of these factors that are needed for an immune response to go to that specific location. So that's how these circulating proteins that are in your blood can end up where there's a pathogen. And so we have C1q that docks to where these antibodies are. There's another complement protein called C2 that'll get cleaved to become C2a and C2b. And then subsequently after that, we'll have another complement protein called C4 that'll get cleaved to become C4a and C4b. Now, this is an important thing. The C2a and C4b will come together to form a unit. It's called the C3 convertase. It has ACE on the end. So what kind of protein is that? Enzyme. There you go. And it says it's a C3 convertase. So it's targeting C3 and it's going to convert it into, I'm sure you guys can imagine based on C2 and C4, it's going to convert it to C3a and C3b. So again, C2 and C4 will come together, will get cleaved. We'll have C2a and C4b that produces the C3 convertase, and C3 will get cleaved. C3b will stick around. C3a is going to be free floating, and it's what's called an anaphylatoxin. It's going to bind to other local immune cells, and it can actually promote anaphylaxis at high enough concentrations. So we're going to hit pause there on the classical pathway. We have also the lectin pathway. So as I mentioned before, there's going to be moments where I'm going to describe how, like a virtual asterisk, where I'll point out how our cells are different than some pathogens. Bacteria have lots of sugar molecules on the surface. One of them is going to be a sugar molecule called mannose. So there's going to be mannose littered across the surface of bacteria. And so we have this large docking protein that's called mannose binding lectin. So it's lectin that binds to mannose here. And so lectin kind of looks like C1q, looks like a spaceship that's docking in. It's going to recognize mannose-rich surfaces. They're going to be typically bacteria. And what's going to end up happening is that it's going to clip C2 into C2a and C2b. I think hopefully this sounds familiar. It's going to cleave C4 into C4a and C4b. We'll have, and then the formation of the C3 convertase, where C3 can be cleaved to C3a and C3b. I'm also going to hit pause there. C3b, so now both of these, if you haven't picked up on it, both of these pathways converge at the formation of the C3 convertase. All right. So if we want to focus on big picture here, main distinction is that the classical pathway involves the binding of antibodies to the surface of the pathogen that kicks off the complement pathway. It kicks off the classical complement pathway. The lectin pathway is kicked off by mannose binding lectin docking in, and both of them together converge at the formation of the C3 convertase. When we get C3a and C3b, C3a is the anaphylatoxin. It's going to be free floating. Certain immune cells have receptors for C3a, and it's going to trigger an anaphylactic response in some cases. C3b is going to be wedged into the surface of that pathogen. And what's really interesting is that for the alternative pathway, C3b is going to actually bind to something called endotoxin. It's this feature on certain classes of bacteria. Whenever you see drugs or implants that are marketed, they're going to say they're endotoxin free. It's meant to try and identify that there's no bacterial infection or not infection, bacteria present on whatever this implant or drug is. And so what happens here is that C3b will actually sometimes bind to endotoxin. It'll actually promote the emergence of more C3b. And so again, you can see all of this is converging at C3b. On the what's going to end up happening is that it's going to promote a few different outcomes here. All right. So there's enzymatic byproducts here of complement. We've talked about C3a, that's an anaphylatoxin. The C3b is actually going to promote C5 to come in, and it'll be cleaved. I'm sure you guys can guess what it's going to be cleaved into. C5a and C5b. The nice thing about complement is you can predict what's going to happen. Both C3A and C5A are anaphylatoxins. And so they're going to actually promote the recruitment also of phagocytes to the site of infection. What's also noteworthy is that the littering of C3b on the surface of this pathogen is actually, now that phagocytes have been recruited by C3A and C5A, C3b is actually going to be a cue for phagocytes to eat this pathogen now. So the first thing is going to be the recruitment of a phagocyte. The next thing is going to be, and these are all like, what are the main outcomes of complement? The recruitment of phagocytes by these enzymatic byproducts, the activation and promotion of actual like engulfing by the phagocytes. The last thing is actually pretty cool, is going to be where complement is completed by the formation of what's called the membrane attack complex, or also called MAC. What's going to end up happening is that C5, when it's cleaved to C5A and C5B, it's going to promote the recruitment of C6, C7, C8. And then once C9 comes in, it's basically going to dig a hole. It's going to drop in and dig a hole basically in the surface of the pathogen. And that's going to create a change in the osmotic pressure, and it's basically going to destroy and lyse this pathogen. So again, the three key takeaways is the recruitment of phagocytes by these anaphylatoxins, by C3b promoting the activation, promote engulfment, and then finally, the formation of the MAC complex to finally destroy the pathogen. I'm going to stop there for any questions. Yes.
Are there three ways of like the ones, are they for all bacteria, or some like one bacteria?
That's a great question. So this is, so complement is not specific to different kinds of bacteria, but literally where I'm going to go next is going to talk about how we have different cells that respond to different bacteria based on the patterns on their surface. So that's a great question. That's actually where we're going to go next. Yes. Yeah, so largely the alternative pathway, basically what happens is as C3b is continuing to be wedged into the surface of the pathogen, it's actually going to bind with endotoxin and actually promote further activation of our innate immune cells. So it's just kind of leveraging a feature of the pathogen to promote further activation. Yes. So endotoxin is a feature on the surface of a certain class of immune cells. So, or sorry, not immune cells, forgive me, a certain class of bacteria. So to maybe go back to your point here, there's going to be receptors where we talk about certain responses to bacteria. The alternative pathway typically is only going to be helpful with a certain class of bacteria. This may be something you guys have already heard of before, like gram-negative versus gram-positive. Endotoxin is going to be present on gram-negative bacteria. And so the alternative pathway is largely going to be most applicable there, because that's the instance in which C3b can bind to endotoxin. It's a combination of like a lipid and like multi-sugar units that that our cells have a receptor for, that promotes wild amounts of activation. And so it just kind of like is another way of triggering further activation by our immune cells. Yes. It's similar in that it is a alphanumeric soup that has multiple pathways, but totally different outcomes. Like this is in response to some sort of foreign pathogen, whereas that's going to be trying to promote hemostasis. Yeah, sorry, the pattern, yeah, sure. All right, I'm going to go one last question, then we're going to keep moving. Yeah. Great question. I actually should have clarified that. We oftentimes associate anaphylaxis with some sort of allergic response. There are different kinds of anaphylaxis that can occur. So like the key features of anaphylaxis typically are the key features of anaphylaxis are going to be drop in body temp, drop in blood pressure. I'm trying to think of the other key things. But that's, so like those are things that are often tied to a severe allergic response, but it's not exclusive to that. Anaphylaxis can happen in response to other things, and at high enough concentrations can happen in response to this. So that's like broad anaphylaxis. There are multiple things that can promote anaphylaxis. One of the most common is going into allergic anaphylaxis. Great question. I appreciate you asking that. I'm going to press on, and I'll try and make sure I stop for more questions shortly. So I've talked about how there are certain patterns or features on the surface of pathogens that our innate immune system recognizes. There's going to be kind of two classes of these patterns, basically. We have pathogen-associated molecular patterns, or called PAMPs. Again, they're pathogen-associated, so they're going to be coming and emerging from some sort of foreign invading pathogen. We also have damage-associated molecular patterns, they're called DAMPs. They emerge in response to tissue injury. You can kind of think of DAMPs, a bit of an analogy I like is you can think of DAMPs as almost as a flare gun to try and alert the body that there's a tissue injury going on. And what PAMPs and DAMPs are recognized by are called pattern recognition receptors, or PRRs.
DAMPs associated only with infectious sources of inflammation?
No, so DAMPs, great question. So for example, you can think of DAMPs in response to some sort of you can think of even surgery. There's a surgical, there's a DAMP. There are DAMPs that are released in response to some sort of large bulk injury. There are DAMPs that are released in response to, even when you get a bruise, there are DAMPs that are released at a small level there. So it's not necessarily restricted to some sort of infectious invading pathogen, but rather oftentimes, and I'm a little biased here because like I tried to do a lot of work with specific DAMPs in like a tissue injury or tissue remodeling context. Does that help answer the question? Yeah. So again, PAMPs are specific features on foreign pathogens. DAMPs are coming from our own cells that are released in response to some sort of tissue injury or cell damage. As an example, ATP is not something that you should typically see just floating around freely outside of cells, but if it is available in the extracellular, the outside the cell environment, that's oftentimes an indicator of tissue damage. Both uric acid and oftentimes cytokines are other well-studied DAMPs. And again, the pattern recognition receptors are what recognize PAMPs and DAMPs. They can actually trigger a few different immune responses or innate immune cell responses here. One of the things they can promote is chemotaxis. That is the migration and movement of cells towards something. So the cells sometimes will actually recognize PAMPs or DAMPs and move to the source. There's a gradient. If you guys watched the pre-lecture, I'm assuming everyone did faithfully as soon as it was posted. There's actually a really unique video of a neutrophil moving across a gradient of some sort of chemoattractant. That's what chemotaxis is, moving towards the migration and movement of cells towards something. These pattern recognition receptors can also help promote antipathogen defenses. They can promote the release of cytokines and chemokines. Cytokines are these cell signaling proteins that oftentimes trigger some sort of response by immune cells. We historically thought that cytokines were only released by immune cells, but over time we've found that other cell populations release cytokines and can communicate with immune cells using them. Chemokines are actually not cells. They're proteins that can promote cell migration. So chemokines are what promote chemotaxis. Chemokines are something that is a signaling protein that promotes cell migration. And finally, all of this can also trigger changes in gene expression, changes in transcription. These pattern recognition receptors can actually be extracellular or intracellular, depending on what they recognize and where we would expect to find that molecular pattern. So we're going to go over a few classic pattern recognition receptors here. One is called formal peptide receptor, or FPR. It's a class of G protein coupled receptors, and soon we're actually going to talk about. We're going to have a lecture on G protein. So if you think yourself, I have no idea what that means, Good, but we're not there yet, and that's okay. So they actually recognize specifically formulated peptides from bacteria, specifically formulated methionine. So one thing we haven't talked about yet is whenever we have gene expression, whenever a gene is expressed, oftentimes there's a specific signal that promotes, that initiates that transcription or that gene to be expressed. That's called the start codon. We're going to talk about that when we get to transcription. But the reason why I'm saying that is because that codon, that start codon, encodes for an amino acid, methionine. In our cells, it's not formulated. Again, virtual asterisk here of how our cells are different than the pathogens. In bacteria, methionine is formulated. So these formal peptide receptors recognize formulated methionine with a couple amino acids that follow it. And so it is a unique way how our innate immune cells can distinguish our self from some sort of foreign pathogen. So the activation of these formal peptide receptors, FPRs, are really great at promoting chemotaxis in neutrophils and macrophages. And what's interesting, we oftentimes talk about humans being a higher order organism than, say, rodents, yeast, bacteria. But what's interesting here is that humans only have 3 FPRs, and mice actually have more. They have 6. Thinking about how things are selected for over time and that FPRs recognize bacteria, why might you think that humans only have 3 and mice have 6? Say that again, sorry? Yeah, mice like hanging out in the trash. So they need a more equipped immune system to withstand that barrage of bacteria, so it doesn't promote some sort of infection that would then kill them. And so over time, like you can imagine that over generations that there have been mice that have been selected for that actually have more FPRs than their peers. And so that's why typically mice actually have, in one instance here, their immune system is unique in that they have 6 FPRs and we only have 3. Again, there's the formal group on the methionine amino acid that is unique to bacteria that we don't have. Another class of pattern recognition receptors are called toll-like receptors. The name toll, so we understood why it's called formal peptide for the previous slide. These are called toll-like receptors because these genes were actually first discovered in flies. And the Latin name for flies are toll. And so when they were also discovered in humans, the immunologist who discovered it got the naming rights and said, well, these are just toll-like. So that's why they're called toll-like receptors or TLRs. And so these are oftentimes going to be localized specifically either intracellularly or on the cell surface. Again, depending on what molecular pattern they recognize, because that would determine where we'd actually expect to find that specific pathogen. And so there's a whole host of molecular patterns that toll-like receptors recognize. We have double-stranded RNA. Again, virtual asterisk here. Our RNA is not double-stranded. We have single-stranded RNA. So if there's any double-stranded RNA that our cells recognize, it would not be from us. So that is a ligand that our TLRs recognize. We have flagellin. That's, again, only available on bacteria. LPS, which is another name for endotoxin, lipopolysaccharide. That's going to be found on the surface of gram-negative bacteria. Gram-positive bacteria have lipid-teichoic acid. So we have both classes of bacteria there, as well as unique CpG islands. CpG is just cytosine and guanine. Those are 2 of our nucleic acids. There are regions of the DNA that have a lot of cytosine guanine bases. And so those areas are called CpG islands. And so we'll talk shortly about why that would be an appropriate ligand or a molecular pattern for our immune cells to recognize. So these toll-like receptors, again, based on what they recognize, they'll either be present on the cell membrane or actually within the endosome. And so for this example, unmethylated CpG DNA, which would be only found in bacteria, which has been engulfed, we would only have access to their DNA if the bacteria has been destroyed releasing their DNA content. And so that would be, that is why the toll-like receptor that recognizes the bacterial DNA is in an endosome where the bacteria would be destroyed and their DNA contents would be released. I'm going to hit pause here and give space for a couple of questions. Are there any questions so far? Yeah. Yeah, that's a great question. Typically damps are going to be unique to that environment because the cells there that respond to those damps are going to be uniquely poised to respond to those damps. Hope that makes sense. So like basically the cell, like there are some damps that are pretty ubiquitous, like ATP is a damp that you could find anywhere. There's some damps that are going to be specific to locations, or at least what they trigger might be tissue or organ specific as well. Are there any other questions? I think we're making good on time. So if you guys want a second for questions, or at least to think of a question. Yeah, go ahead. Can you say that one more time? Sorry. How do these receptors interact with other parts of the immune system? So they're not they're interacting with components that are inside the bacteria that have been released. So like the endosome is something that can when something is endocytosed, that means it's in an endosome. Over time, that endosome matures into something called a lysosome, which means it lyses, it's dropped the pH a ton, and that drop in pH, again, as we talked about amino acids and proteins, change in pH or change in charge can unravel or lyse something. And so the contents of the bacteria are released. And so if there's a unique molecular pattern inside that bacteria that our cells recognize, those receptors are going to be in the endosome, so it can respond right away. And so those receptors, even though they're inside our cell, they can trigger downstream consequences that will promote either cytokines to be released, chemokines to be released, genes to be expressed. And so even though those receptors are not on the surface of the cell, but rather inside in the endosome, it can trigger a lot of downstream events. Does that help answer your question? Some of these pattern recognition receptors are inside our own cells, yes. So these patterns, so okay, that question helps. Our pattern recognition receptors typically are either on our cell surface, on the cell membrane, or can be intracellular. So one example is toll-like receptors that can be within our endosomes that specifically target components of bacteria that have been released within the endosome. Does that help clarify? Great. Saw one more question over here. Yeah, so this is one that I added in that is meant to just illustrate where these toll-like receptors can actually be situated. So I added it in because it felt like on the previous slide, it was, I think, hard to appreciate from a list where some of these receptors can be. And so the main takeaway here is just that these receptors can be situated not just on the cell membrane. Yeah. No, so I'm not gonna, so great question. So I'm not gonna ask you to remember all of the TLRs. The ones I've boxed in, these four, TLR2 is technically in the box. So imagine TLR2 is boxed in. TLR2, 3, 4, 5, and 9 are ones that I'm gonna want you to know and what they bind to. Not the entire list. Does that help? All right. Once the screen comes back on, I'm going to move on. So one of the pattern recognition receptors, or one of the molecular patterns that we've talked about a couple times now, is lipopolysaccharide, or LPS. This is what is also known as endotoxin. We talked about it a few times here, but basically with LPS, it's going to be found particularly on gram-negative bacteria. and I'm going to ask you guys to remember that. It's basically a combination of a lipid tail with a core oligosaccharide. And this is a really potent activator of different innate immune cells. Sometimes the smallest amount can trigger a really strong inflammatory response. And so this is going to be a key factor to remember. And this is oftentimes associated with binding TLR4. So when we talk about gram-negative bacteria, there was a scientist named Graham who wanted to try and distinguish between certain classes of bacteria. So he developed a stain that binds particularly to peptidoglycan. It's this peptide sugar molecule that is represented in the blue structure. So if this Gram stain binds to peptidoglycan, what it's going to be able to do is it's going to be able to bind to bacteria that have the exposed peptidoglycan on the very surface of these bacteria. Whereas in some bacteria, there's going to be an outer and an inner membrane, excuse me, and peptidoglycan is actually wedged between them, and it's going to be inaccessible by the stain. So gram-negative bacteria are going to have the shielded peptidoglycan that is going to have lipopolysaccharide or LPS on the outer membrane. And the gram-positive is going to have peptidoglycan on the outer surface, as well as a couple key molecules that also characterize gram-positive bacteria. One of the other key molecules that identifies gram-positive bacteria is something that's called lipoteichoic acid, also known as LTA. It's this combination of a glycerophosphate with several sugar molecules, but in the graphics to the right, you can appreciate what lipoteichoic acid looks like in the context of that peptidoglycan region for gram-positive bacteria. We talked about this really briefly, but we also have another molecular patterns, cytosine, phosphate, guanine. So cytosine and guanine are nucleic acids. We have we have lots of regions within our DNA that are rich in CG regions. They're called CPG islands. They're separated by a phosphate group here. What's important to note is that many of our CPG islands, again, regions that are rich with cytosine and guanine, are methylated. They have methyl groups added there. Bacteria do not. So there's typically you'll find unmethylated CPG islands specifically in bacteria. What I should say is that there is like regulation of methylation of our DNA. And so that's not to say like you'll never find unmethylated CPG islands within humans, but typically they're methylated. And so it's TLR9 that binds to these unmethylated CPG islands. And again, that the bacterial DNA will be made available within the endosome. So the TLR for unmethylated CPG islands is going to be found within the endosome. There's actually a scientist here at UVA that's in our immunology department that was one of the key people that helped discover what TLR9 actually binds to. And so, a lot of immunology, some of these cells were discovered back in 1905 or even 1898, I believe, but there's still a lot of work that's being undertaken to identify a lot of these things and characterize these things in new ways. And so, yeah, there's a methyl group that's on the cytosine there. The second to last pattern recognition receptor that's intracellular that we're going to focus on is called cGAS. So cGAS, what's unique about this one is that it senses cytoplasmic DNA. So as you can imagine, our DNA is not free floating within the cytoplasm. Our DNA is restricted to the nucleus. So cytoplasmic DNA is typically a signal that there's some sort of other pathogen whose DNA is present. And so cGAS binds to cytoplasmic DNA, and it can actually generate a molecule that's called cyclic GAMP. So it's actually taking GTP and ATP. They're each losing, it's triphosphate, so they're losing 2 phosphate groups, and they're going to be cyclicalized to one another, meaning that they're going to be bound to one another across their sugar molecules, and that's how we get cyclic GAMP. Cyclic GAMP is going to go ahead and activate another molecule called STING, which then STING promotes the expression of a class of proteins called interferon. So interferon is really key in our immune system because what ends up happening is that if there's some cell that has cytoplasmic DNA within it, that activates cGAS, that then generates cyclic AMP that activates STING. Interferon is going to lead every cell in the surrounding area to express a certain protein. Every cell in our body has a protein that's unique to you and only you. That's what's really key for organ transplants, where there has to be tissue matching, and it's this specific protein. You can think of it almost as like as an ID card for every cell in your body, every nucleated cell in your body. And so your protein is going to look different than mine. Interferon prompts all the surrounding cells to express that protein at even higher levels so that our immune cells can identify who's not expressing this ID protein. It's called MHC class 1. And the cells that are not expressing it are typically ones that are either going to be virally infected or have some other underlying issue. And so interferon is a way for our cells, our innate immune cells, to identify infected cells based on their expression of this ID protein, this MHC class 1. The final pattern recognition receptor we're going to talk about is one that's called NOD. We have NOD1 and NOD2. Typically, this receptor typically recognizes a certain peptidoglycans or a certain sugar molecule that's with a peptide. What NOD2 specifically, what it's going to bind to is something that's called myramyl dipeptide. So it's going to have, let me move my mouse. So we have N-acetylmyramic acid up top, and we're going to have two amino acids down below. That's the dipeptide part. So NOD2 is going to specifically recognize that certain molecular pattern, and it's going to be specifically and the receptor that binds this molecular pattern is usually going to be found in monocytes, macrophages, as well as gut epithelia. That's going to be key because we usually find this molecular pattern, myramyl dipeptide, within gram-positive bacteria, and so that means that's going to be oftentimes a classification bacteria that you won't typically want to see within the gut because it's going to trigger a lot of NOD2 activation. But what's really interesting is that NOD2 is what's been implicated within Crohn's disease. NOD2 is what's called a susceptibility gene. It means that it's a gene that's been determined to have certain mutations that can raise the likelihood of an individual developing the disease. And what's really fascinating is that back in 2001, there was so there's the three biggest journals in the world are Nature, Cell, and Science. We're all trying to publish in that, which would be really cool, but it's a real shot in the dark for all of us, more so myself than anyone else. What was really cool is that there was an issue in Nature back in 2001 where two independent studies identified something about Crohn's disease that corroborated one another, and they actually published them in the same issue and as back-to-back articles. One study was called a linkage analysis. What that does is it looks across the genome or across our DNA and tries to identify mutations that are linked to a specific disease or disease outcome. And so this one study, what it did was it identified chromosome 16 as what's called a susceptibility locus. Locus just means specific spot in our DNA or within our genome. And so it identified that specific spot, chromosome 16, especially based on this graphic here, the peaks are where the susceptibility locus was highest, or the linkage analysis was highest. But bless you. Those regions of the chromosome 16, this locus here, had the highest susceptibility to Crohn's disease. What's interesting is that you can imagine, perhaps, based on the title of this slide, what gene is at that locus. And it's NOD2. The subsequent study demonstrated that if NOD2 is truncated, which means if it's shortened, that it's actually truncated within some patients of Crohn's disease, and that leads to a loss of function. Loss of function means that it just doesn't work anymore. So in Crohn's disease, it seems like some patients have a truncated NOD2. This receptor some of it is chopped off, and that leads to NOD2 not working anymore. What's really interesting is that they actually did this within a family study where the white square circle are mom and dad that are both heterozygous for this truncated form NOD2, which means they have both the normal copy, I shouldn't say normal, but the quote unquote normal copy of NOD2 and this truncated version. They're each a carrier for one copy of it. And that's demonstrated here in this DNA gel, where lane 1 and lane 4 are mom and dad, and they have the wild type allele, the wild type copy of NOD2, and the truncated version. And their subsequent progeny, their children, both inherited both truncated copies. So they're homozygous for the truncated version, which led to the manifestation of the disease in the children. So what ended up happening is that there was a subsequent study a few years later that took that truncated version of NOD2 and knocked it into a mouse, which means they genetically engineered the mouse, for lack of a better phrase, where they took they were able to insert that copy of the truncated NOD2 into the blastocyst of a fertilized embryo, and actually generate a mouse that has only the truncated copy, and looked at that and compared it to a wild type mouse, or a mouse that doesn't have the truncated copy, and used a certain chemical to trigger colitis. What they were able to see, based on this histological image that just disappeared for a second, but momentarily will come back, so bear with me, bless you. The idea is that just by inserting this truncated copy, they were able to replicate the full pattern of the disease demonstrated by at least this is a model of colitis, I should say. So here they're able to lose the entire mucosal layer of the GI tract there, and that the similar results were obtained by completely knocking out NOD2. So just having the truncated copy was functionally the same as losing the entire gene. And so this was a great way to try and confirm what was demonstrated in those previous studies published in Nature. For treatment for Crohn's disease, oftentimes patients can take corticosteroids, but that's oftentimes dealing with the symptoms. One of the most commonly used drugs for Crohn's disease is antibodies that neutralize tumor necrosis factor, or TNF. That's one of the most inflammatory cytokines that we have in our body. And so inhibiting it, neutralizing it with an antibody that targets it, seems to work pretty well in trying to dampen the inflammatory nature of Crohn's disease. You might recognize one of these neutralizing antibodies, Humira. It is one of the top drugs on the market right now. And so it's it's also used for other instances beyond Crohn's disease, like rheumatoid arthritis, as well as a few other applications as well. I'm gonna hit pause here and stop for a couple of questions. Do I have any questions about what we've talked about so far? Yeah. Yeah, great question. So a knockout mouse means that the entire gene is based, or either the entire gene or certain exons, like certain, like the actual coding parts of the DNA of the gene, are taken out. Knock in typically means we're actually going to go in and edit and change a certain nucleotide or a certain region. And so in this case, they were able to knock in or put in a truncated copy as opposed to just getting rid of the entire coding region of the gene. Bless you. Yes. Oh, I skipped that only because we typically would have another lecture before this that would provide the context of what a DAG anchor. It's just diacylglycerol, which is a molecule that wedges a molecule within the surface at the surface membrane. When we get to G proteins, that'll make a little more sense. We usually have that lecture before this one, but we tried to move it around, and so it's not something that you'll be quizzed on because we haven't touched on it yet, but we'll get to that when we get to G proteins. Thanks for catching that. I should have actually pointed that out. All right, so if any of this really fascinated you, whether it was innate immunity or Crohn's disease, we actually have a really strong molecular immunology department here. Upcoming AO looks at the combination of neuroscience and immunology. Amy Bouton does some really great work on infectious disease, as does Melanie Rakowski, looking at infectious disease as it relates to cancer. Sapita Dolasahi is from our department and does really great work using systems biology to understand our immune system. And two scientists that I really respect outside of our community that have done a lot of great work in this space are Dabrowski Herbert at UPenn and Irene Salinas at University of New Mexico. So if you have any questions, we'll be hanging back here. Thanks for your time, guys.